Metabolism PhysioLab platform is a mechanistic, mathematical model of whole-body human metabolism and physiology that is used to understand and evaluate type 2 diabetes, obesity, and treatment approaches for each. The Metabolism PhysioLab includes the metabolic and physiologic pathways associated with glucose and lipid metabolism, including key participants in adipose, liver, muscle, small intestine, pancreas, stomach, circulation, and brain. Virtual patients provide the ability to use the Metabolism PhysioLab to test different hypotheses, as each virtual patient has a unique combination of underlying metabolic lesions. Virtual patients include differences in the underlying metabolic and physiologic pathways that are consistent with what has been reported in the literature. Type 2 diabetes is a heterogeneous disease with a multitude of mechanistic and clinical differences between patients. The virtual patients within the Metabolism PhysioLab capture this broad range of diversity and represent patients that are lean, overweight or obese, healthy, pre-diabetic or diabetic, with varying degrees of severity. Each virtual patient appropriately responds to a variety of short and long-term clinical and nutritional protocols, including fasting, oral glucose tolerance tests, and infusions of nutrients and hormones. Exercise is also included, providing the ability to explore changes in long-term energy and nutrient balance and subsequent effects on body weight. In addition to properly representing the pathophysiology of type 2 diabetes and obesity, standards of care treatments for each are also represented within the platform. Simulations of these treatments can be performed as monotherapies or in combination. The Metabolism PhysioLab also includes the ability to represent novel compounds in clinical development, as many of the current targets for type 2 diabetes and obesity are represented in the platform. The pharmacokinetics and pharmacodynamics of a compound against one of these or other targets can be represented based on proprietary data, and the resultant changes to the target and downstream improvements in pathophysiology can be predicted. Clinical protocols, such as an oral glucose tolerance test and hyperinsulinemic clamps, can be simulated to identify measures to include in preclinical studies and clinical trials to demonstrate efficacy. These novel compounds can also be simulated as monotherapies or in combination with standards of care. Entelos has partnered with a number of major pharmaceutical companies to apply the Metabolism PhysioLab to identify and validate targets, identify optimal characteristics for compounds, identify optimal dosing protocols and measurement sampling times, optimize clinical trial protocols, and identify responsive and non-responsive patient types. The Metabolism PhysioLab platform predictions have been tested against a number of key clinical data sets, such as those listed here from clinical trials with treatments for type 2 diabetes. The Metabolism PhysioLab platform includes representations of the primary standards of care for type 2 diabetes, including metformin, pioglitazone, rosiglitazone, sitagliptin, exenatide, injected insulin, glyburide, glimepiride, neteglanide, and acarbose. Virtual patients in the Metabolism PhysioLab platform have appropriate changes in common primary and secondary endpoints, such as hemoglobin A1C, fasting plasma glucose, body weight, and triglycerides when simulations of these standards of care are performed. Simulated monotherapy and combination treatments can also be performed on the virtual patients with these standards of care. An example of the predicted response for virtual type 2 diabetes patients on the standard of care treatment metformin is shown here. The predicted clinical responses to metformin agree with measured data reported in the literature. On the right is a figure comparing the change in hemoglobin A1C over 24 weeks, with the measured data displayed in black and the simulation results in red. There is good agreement between the predicted changes in A1C over 24 weeks and the measured data. On the left are two figures illustrating the diverse range in responses to metformin amongst the myriad virtual patients. The figure at the top also shows the change in A1C after 24 weeks of treatment with metformin. As it is well documented that the change in A1C is dependent upon the initial pretreatment A1C, we have plotted the data in this way. The blue diamonds in this figure are from various clinical studies reported in the literature. See references at the bottom. The yellow squares are individual virtual patients. The general relationship between initial A1C and change in A1C from the measured data is captured by the virtual patients, validating the representation of metformin in the Metabolism PhysioLab platform. There is also a representative amount of variability amongst the virtual patients. The bottom figure is analogous to the top, only the change in fasting plasma glucose is related to the initial fasting plasma glucose. Again, the relationship between initial and the change in fasting plasma glucose is consistent between the measured data and the simulation results. This provides further validation of the representation of metformin in the Metabolism PhysioLab platform. Similar validations have been performed for pioglitazone, rosiglitazone, sitagliptin, exenatide, injected insulin, glyburide, glimepiride, nateglinide, and acarbose. For more information, please contact Intellos.